immunology, and our research team who are all involved in this project is it is collaboration of three universities: University of Skövde, Örebro, and Gothenburg. And in Skövde, Patrick Nilsson, Anna Karin, and Andreas Johnson are involved. And Örebro, Elizabeth and Elgarbet. Gothenburg, Yuyan, Maria, and other master students like me, Hala. And coming to, thanks to all the people who are involved in theory as well as lab sessions, because without them we cannot, we would have not done master's degree. And and my special thanks to Anna Karen because of her supervision, and I am able to do independent research because of Anna, and my thanks to Hala also for contributing and all other friends. And this is about my project.
Thank you. Maybe we should have a look at your poster before we... Any comments on the poster? My personal comment, I think it's a really, really good poster.
Thank you.
Several reasons. There's a good balance between text and focus on project and results and the methods. Maybe you could have skipped the positive and negative selection part. Maybe also you could highlight the conclusions a bit more.
Oh.
I think the balance is perfect and it's very easy to read. Also good to have a photo of yourself.
Anna said me to include the photo.
Yeah. Usually, I mean, poster is a way to communicate science on a conference.
Yeah.
And there you have poster sessions, and we have several hundreds of posters in the same area. And then it's always easy to find the person who is responsible for the poster.
Okay.
Usually we want to discuss the methods or results.
Yes.
It's easier to have a photo.
Thank you.
What is also a good idea if you are presenting a poster is to have handouts. Maybe you could, I think you can make a small book or I mean, the A4 format is pretty good, but you can make it a bit smaller when you...
Okay.
It's really good to people to take a copy, because after a while, several hundreds of posters, you get really fed up with reading all the posters. Then you can take one copy to a hotel room and read it in the evening. Any other comments?
Questions? Any questions?
We can start with this is I think, as an examiner, I think in general it's very well written. I think.
Just please.
This is. But I think there is one big problem with thesis, and which you need to revise. And before I go into details, I just want to mention one thing. When we examine your thesis, of course the results are important, but they are not crucial for the grade. So what we are actually looking at is how you communicate science. And one important part of the thesis is, of course, the report is how you communicate results in the discussion section. And as it is now, basically you have no reference whatsoever. You tried, I think it was a good idea to combine results and discussion into one section. sometimes it's easy to write. Write a report then, but you need to put your results into context, into a scientific context.
Okay.
And you do that by referring to other papers. The problem is sometimes is when you don't have enough results. Like in your case, all the results seem a bit trivial or basic. What you could do then is to, of course you could discuss your results to some extent, but then you could focus more on discussing the methods and put your methods into the scientific context. And that is, I think, what you should do in this report. Focus more about the methods you have used.
So, like in the results and discussion part?
Yeah. I think you should then, as I say, you need to put your results or your methods into context, scientific context, and you do that by referring to other papers. You can do that with respect to your results or your methods. In this case, you can do both. But I think it will be easier to focus a bit more on your methods. Because it's not that many. How many papers did you find on beta track? Not many. One or?
Very few papers about the beta track. Alpha track we find a lot of.
Yes, but you can also, of course, you can discuss your results or your methods in relation to work done on the alpha track.
Okay.
I think that's the only thing you need to realize. Of course, it's not a small task, but as I said, it's very well written. The only thing maybe some of the figures or tables could be combined to, for example, table 1, 2, and 3. To me, it seems that they could actually be, we could combine them into one table instead of 3 different. But it's just a matter of taste. I think it's not necessary. Do you have any comments?
No, that was also my major comments from me.
Yes. During the correction also unnecessary to consider.
Any questions? Or?
I will ask one question. Why is a beta track good to look into if we want to see how thymocytes are developing? Why can't we look at the genomic sequence? Why do we look at the beta track?
In order to?
We are looking at the thymocytes, how they are developing, how they are dividing and so on.
Okay.
And so why do we look at the beta track then? Why is it good to look at beta track when cells are dividing?
Yeah, that's a good question. So, because in order to know whether the thymus function is proper or not, we have to look for the all different thymocytes which are present in the thymus. Like we have to look at the all stages of thymopoiesis, like double negative, double positive, and single positive. So, if in some cases where there are abnormalities in the thymic function, so accelerated transition may take place. Suppose may from double negative to single positive. So in that case, we have to look for the tricks, because tricks are present between the double negative and double positives. So that is the main reason we are looking for the tricks, not genomic.
Yeah, and the tracks are just diluted.
They are diluted.
They're not
Yeah.
copied.
Because during the mitosis of thymocytes, they will not be replicated. So their frequency is diluted.
Also important to highlight. Thank you.
Yeah, thank you.
Thank
It
you.
was a very good presentation.
Thank you.
Syns vi alltid, eller halv 11?
Ja,
Ja, jättebra.